get my slides up. All right, so just to confirm that you can see my slides.
Yes, we can see them.
Thank you. All right. So hi, everyone. I wanted to first thank the Stevens for the invite and for all of you for joining. I'm really excited to share with you a project that, we're kind of just finishing up with the data analysis. We're chewing on what it all means. So if you do have any thoughts about it, feel free to, either ask me at the end or even like email me if you have any pressing thoughts. And it involves analyzing the genomes of nitrogen fixing microbial symbionts. All right, I have to move. Okay. So thanks to the next generation sequencing revolution, it's never been clear that we live in a microbial world. I don't think it's a surprise to anyone here. Humans, plants, and animals are colonized and infected by a plethora of microbes, including bacteria, archaea, fungi, and of course, viruses, which we're hearing a lot about today. And these symbiotic microbes are not just living with us, some can greatly impact our health. So for example, lactobacillus that helps us digest lactose, or the all too relevant novel coronavirus, it's, taken the lives of over 3 million people worldwide in just over a year. So clearly this wide range in effects of these microbes, it becomes critical that we're able to predict how microbes will impact host health. And while microbiome science has begun identifying how microbial community composition impacts the function and the knock-on effects on host health, we lack an evolutionary framework that can be used to predict how beneficial or detrimental these interactions will become over evolutionary time. For example, if coevolution between a host and microbe is driven by selection favoring one partner at the expense of the other, or in other words, fitness conflict, then these interactions will become more detrimental and break down over evolutionary time. Alternatively, if hosts and microbes co-evolve in such a way that selectively favors cooperation or fitness alignment, then these interactions are likely to become more beneficial and stable over evolutionary time. However, host and microbial fitnesses are actually these complex quantitative traits that are governed by many genes. And so fitness alignment at the whole organism level doesn't necessarily preclude conflict at the genomic level. So we need to be able to, in order to predict symbiotic interactions and how they're potentially co-evolving over time, we need to understand the genetic basis of key symbiotic traits and determine the degree of conflict or cooperation that are apparent at both the whole organism and genomic levels. So a well-established approach for predicting how traits evolve generally involves statistically testing for the association between genetic variation, for example, whether an individual possesses a C or a T at locus 7, and phenotypic variation in traits of interest, such as plant yield and agriculture. So in doing so, we're essentially mapping the genetic basis of important phenotypes by building a genotype to phenotype map. In the image to the right, I'm just showing the phenotypic differences between two plant genotypes that grew in the exact same environment. However, you can tell that one of them is doing much better than the other. It's growing larger, producing greener leaves. So by determining how these individuals differ genetically, we can get a better sense of what genetic mechanisms underlie plant growth in this case. But of course, there's a lot of complicating factors. Rather than just going from the straight path from genotype to phenotype, these maps often have quite a few detours, if you will. Or in other words, features that can make our predictions slightly more complicated. So today I'm going to be focusing on one such feature that involves a single gene or genomic variant that affects more than one trait, which is also known as pleiotropy. We heard a little bit about this actually in Dr. Turner's talk. Such pleiotropic variants are especially critical when we want to determine the genetic basis of symbiotic traits, because it's often the case that variants in one individual not only affect multiple traits of that same individual, but also traits of interacting individuals, which I'm going to be referring to as symbiotic pleiotropy. So for example, the phenotypic differences between these plants is not just due to their own genome, but also that of their interacting microbes. While symbiotic pleiotropy can be used to pinpoint the genetic basis of symbiotic traits, it also allows us to quantify the degree to which host and microbial fitnesses are aligned or in conflict, and therefore whether these interactions will remain stable over evolutionary time. At the organismal level, we can determine the degree of conflict or alignment by examining genetic correlations, or the covariance among genetic means for each fitness proxy. Positive genetic correlations would thus imply that host and microbial fitnesses are aligned, while negative genetic correlations would imply that host and microbe fitnesses are in conflict. We might see that strong fitness alignment at the organismal level is governed by many pleiotropic variants whose effects are in the same direction or concordant. So for example, variants associated with both an increase in microbial fitness as well as an increase in host fitness, which is represented by the cloud of points falling to the right of 0. Alternatively, we might see strong fitness conflict at the organismal level that's governed by many pleiotropic variants whose effects are opposite in direction or discordant. So for example, variants associated with an increase in microbial fitness, but a decrease in host fitness, represented by the cloud of points falling to the left of 0. And of course, we might just see a lack of relationship between host and microbial fitness that's governed by variants who are mixed in their effects. So I'm just kind of putting these up here as a primer for the data that I'll be showing you so that we can more easily interpret them. But before we can truly understand symbiotic pleiotropy, we need to have reliable measures of microbial fitness. So for those of us who work on microbes, you know how challenging obtaining these measures can be, especially when we care about microbial fitness within the context of symbiosis, or when we're actually trying to quantify how much fitness benefit microbes are obtaining from the relationship. So in order to examine the genetic basis of symbiotic traits at both the organismal and genomic levels in natural microbial populations, I used the Legume-rhizobium Symbiosis, which has been a model that's been powerful for understanding host microbe coevolution. Legumes provide carbon to rhizobia, which are alpha proteobacteria that reside within nodules located on the plant's roots. And in return, rhizobia are able to fix nitrogen, so they convert atmospheric nitrogen into a plant usable form, which results in this beneficial exchange of resources. But what's really cool about rhizobia is that they often possess these multi-part type genomes. So they often have these large chromosomes as well as one or more symbiosis plasmids that can also be fairly substantial in size. In this example, I'm showing you the 2 symbiosis plasmids characteristic of Ensifer, or formerly Cynorhizobium alleloti, which is the species I've spent a lot of time studying. The symbiosis plasmid B, or PSYMB, contains many genes involved in survival within the rhizosphere. And then on PSYM-A, there are many genes that are canonical for establishing the symbiosis as well as fixing nitrogen. Okay, so to get at whether fitness, conflict, or alignment is more prevalent within natural populations of rhizobia, I utilized data from 2 massive projects. First was a single inoculation experiment conducted in Katie Heath's lab, who's pictured to the left if you don't already all know her. And this involved 191 strains of Ensifer melliloti that was paired with 2 host legume genotypes. And these isolates were originally collected from natural populations spanning Spain to France, which is actually the native range of the legume host Medicago truncatula. So each strain's whole genome was fully sequenced. And then replicates of the 191 isolates were singly inoculated onto one of 2 plant lines. So we had DZA or A17. And then several proxies of rhizobium quality, or the benefit the plants were getting from the relationship with a particular strain, were measured. So we measured chlorophyll A content in the leaves, which is assumed to be related to how much nitrogen rhizobia fix. as well as above ground plant biomass, which is a very common proxy to get at how much the plant is benefiting. We additionally measured proxies for rhizobium fitness, including both nodule number and nodule weight. Now I'm going to talk a little bit more about those in the following slides. The second data set came from an experiment led by Leona Burkhart, who's a former postdoc of Peter Tiffins at the University of Minnesota. And this experiment utilized 89 of the 191 strains of Ensifer, but it involved a mixed inoculation experiment. So this meant that every plant was inoculated with all 89 strains at the same time. So in doing so, we're able to get at an additional measurement of rhizobium fitness, which is the ability of each strain to nodulate the two hosts when in competition with the other. So using the whole genomes for all 191 strains and phenotypic variation measured in both experiments, I conducted a genome-wide association study using a pretty standard pipeline. But briefly, reads were aligned, variants were called, and then filtered for a final set of about 7,500 that passed into the program GEMMA. And what GEMMA essentially does is to identify the genomic variants that were significantly associated with your traits of interest. And so what it outputs is this effect size for each variant that represents both the strength and the direction of the association between that variant and your trait of interest. So we combine the phenotypic and genomic data to first ask, what are the relationships among rhizobium fitness components at both the organismal as well as the genomic levels? Are these traits genetically distinct and therefore evolve independently, or are they linked through pleiotropy? We use this information to next address the potential for genomic conflict in the symbiosis. And we're asking whether variants tend to have concordant or discordant effects on host and symbiote fitness. And then finally, we ask whether there's any evidence that historical selection has shaped genes underlying fitness conflict or alignment differently. And this is what we might predict under different models of coevolution. Okay, so let's get into the results. But I'm going to go through a little bit about what we mean by rhizobium fitness. So we know that rhizobia get a significant boost in fitness when they successfully nodulate plants. And in fact, it's been estimated that a single rhizobium cell that establishes a nodule is thought to gain a fitness benefit of 10 to the 5 or more. So more nodules of the same strain presumably would then be equated to greater rhizobium fitness at the plant level. But we know also that this estimated fitness benefit depends on nodule size. So the larger the size, the more reproductively active cells are then released back into the soil after symbiosis. And when nodule number and size are quantified in these single inoculation experiments, they become really useful because they can be directly correlated with the genotypic mean of partner quality. So how much benefit the plant's getting of individual strains. And that allows us to kind of draw these correlations. Recent advancements, however, that merge quantitative genetics and metagenomics allow us to address what is perhaps the most important component of rhizobium fitness, which is the strain's ability to compete for nodulation. So the first question we're asking is, how do these three different fitness proxies relate to one another? And we're going to examine this again at the organismal and genomic levels. Okay. So here are the results at the organismal level when you regress the strain means for nodule number, which is appearing on the x-axis, and nodule weight on the y-axis. The results for plant line DZA are always going to appear at the top in green, while those for A17 appear at the bottom in pink. So for this particular pairwise regression, we see a significant negative correlation, suggesting that these two fitness proxies are trading off with one another. And so another way of saying that are strains that are forming large nodules tend to form really small, fewer of them, sorry. And then those that form smaller nodules tend to form many of them. Here are the results at the genomic level when we're regressing the effect sizes for nodule number on the x-axis and nodule weight on the y-axis. And again, the effect size represents both the strength and the direction of the association between the trait of interest and a particular variant allele. So just to kind of layer in some information here, because I know there's a lot going on. Each dot represents a variant that is significantly associated with one of the two traits, and those are appearing in color here, whereas the dots shaded in black are actual pleiotrophic variants. So they're significantly associated with both traits of interest. And then at the top left and right corners of each figure are the percentages, representing the proportion of significant variants falling to the right or left of 0. And then the counts within the parentheses are just the total number of pleiotrophic variants in each side. Okay, so from this data, we can see that most significant variants and actually all pleiotrophic variants fall to the left of 0, meaning that their effects are discordant, opposite in sign, or in other words, antagonistic. So antagonistic pleiotropy is, again, something we heard about in a previous talk. So for example, this means that a variant's positive effect on nodule number tends to have a negative effect on nodule weight. Okay, so here are the results when we're regressing strain means for nodule number on the X and competitive fitness on the Y. And we see a significant negative correlation, but only for one of our plant lines, for DZA. Again, suggesting this trade-off, but we actually don't find a correlation for A17. And then here are the results at the genomic level. So for DZA, most of the significant variants are falling to the left of 0, again, suggesting that these effects tend to be antagonistic. Whereas for A17, variants fall in nearly equal proportion on either side of 0, although the majority of those pleiotrophic variants have antagonistic effects. Okay, and then finally, we regress strain means for nodule weight on the X and competitive fitness on the Y, and we see significant positive correlation for DZA only. So this suggests that fitness proxies are positively genetically correlated, whereas we saw a lack of correlation for A17. And then at the genomic level for DZA, most of the significant variants are falling to the right, suggesting that their effects for both fitness proxies are in the same direction, or otherwise they have positive effects, whereas A17, significant variants fall in equal proportion on either side, but the majority of the pleiotrophic variants have antagonistic effects. So this is kind of a cool example where, depending on what plant line you're looking at, you're going to see different results. Okay, so this is just a summary figure that is specifically counting the pleiotrophic variants that had antagonistic effects in bluish gray versus those that had positive effects in orange. So from this, it should be really clear that for most of the fitness proxy combinations, we're seeing this antagonistic pleiotropy. It represented about 90% of all the variants that we looked at. Another thing I want to point out is that the stronger genetic correlations were not necessarily governed by more pleiotrophic variants. As you can see for the regression between nodule number and competitive fitness, which showed the highest number of pleiotrophic variants, but not necessarily the steepest slopes. And then another thing I wanted to point out is that some trait combinations weren't governed by many pleiotrophic variants. So this suggested that these traits are governed by potentially different underlying genetic mechanisms that are likely to evolve independently. So this is particularly the case for competitive fitness and nodule weight. So to summarize this section, we found nodule number tended to trade off with both competitive fitness and nodule weight, the latter two of which were positively correlated, but only on one of the two host lines. By examining variant effects, we overwhelmingly found that these traits are governed by antagonistic pleiotropy, suggesting that in order to maximize fitness, rhizobia are evolutionarily constrained into these two categories. So they can form larger nodules and be more competitively fit, or form numerous small nodules. And as we'll see in the following slides, this latter strategy is likely associated with fixing less nitrogen as well. So let's move on to our next question. Are host and symbiote fitnesses aligned, conflicting, or unrelated? Here are the results when we regressed shoot biomass, which is our proxy for plant fitness here, and one of the rhizobium fitness proxies, which is nodule weight. So you could see that on both of our host lines, our fitness proxies were positively genetically correlated, suggesting that fitness alignment seems to be prevalent, at least for this particular trait pair. And here are the results at the genomic level. So you can see many of the variant effects are falling to the right of 0, suggesting that the positive genetic correlations were mostly driven by variants with concordant effects. So a variant that's associated with an increase in plant fitness tends to be associated with an increase in microbial fitness. When we regressed shoot biomass and competitive fitness, however, we found a positive correlation when traits were measured on DZA, but actually no correlation when they were measured on A17. And interestingly, at the genomic level, variants were mixed in their effects, whereby almost an equal proportion fell to the right or left of 0, despite the positive correlation at the organismal level for DZA. I guess it's important to note, though, that most pleiotropic variants did fall to the right of 0, suggesting that we are seeing a genomic signature of alignment overall. And then finally, when we regressed shoot biomass and nodule number, we found a complete lack of correlation for both hosts. And at the genomic level, while most variant effects fell to the right of 0 on DZA, suggesting greater alignment, the opposite was true for A17, with most variants falling to the left of 0, suggesting greater conflict. It's important to note that when inconsistencies between the organismal level and the genomic level patterns emerge, such as that seen here, whereby we see a lack of correlation at the organismal level, while having many concordant effects at the genomic level, these inconsistencies are due to how different alleles are actually packaged into the individual genotypes, which then determines the genetic correlations observed. So, it's not always the case that the two levels are going to mirror one another. And this packaging is shaped by many factors, including population structure and linkage disequilibrium. So these are things we could actually go and look into more detail to see why there's a disconnect between the two levels. Okay, so here, just like before, I'm tallying all the numbers of pleiotropic variants, underlying fitness conflict or alignment. And basically you see more orange in this plot, right? So 80% of the total pleiotropic variants that we looked at seem to underlie fitness alignment. And most of the pleiotropic variants were associated with rhizobium quality and nodule weight. So these results suggest that fitness alignment seems to prevail in natural populations, which is actually not what we expect based on theory within mutualism, where you expect more conflict between the two partners. We nonetheless, though, did find some loci underlying conflict, and this was particularly true on A17, in which many pleiotropic variants were negatively associated with shoot biomass, but positively associated with nodule number. So follow-up functional validation studies could then be done, because we know where these variants occur, and so they can be used to understand how these candidates of conflict potentially disrupt the Legume-rhizobium symbiosis. Taken together, our results suggest that fitness alignment for at least 2 of our 3 fitness proxies, namely nodule weight and competitive fitness, are pretty much in alignment with host fitness, and that variant effects underlying these traits are also mostly in alignment. When we actually go back and take into account the relationship among rhizobia fitness proxies, we would predict that selection should favor strains that are forming larger nodules and are more competitively fit, that also confer greater benefits to the host. But it's really important to point out that differences between our hosts were observed, and some of these differences might actually be due to important traits that differ between the hosts. For example, their ability to reward more beneficial rhizobia. And we predict that in the case of a non-choosy host, for example, you might actually reveal much more of these mechanisms of conflict that potentially could be detrimental to host fitness over time, unless hosts evolve counter-selection mechanisms. Okay, so briefly, I'm going to walk you through some preliminary results that involved examining patterns of selection across genes that underlie conflict versus alignment. So what we've done so far is we calculated 2 commonly used test statistics, namely pi, which is on the left, that represents average pairwise nucleotide diversity among our 191 strains. while Tajima's D was calculated on the right, which represents the difference between two measures of genetic diversity, which is the mean number of pairwise differences and the number of segregating sites. We calculated these statistics for 4 categories of genes. So those underlying rhizobium fitness pleiotropy, namely positive and antagonistic pleiotropic genes, as well as genes underlying symbiotic pleiotropy, namely alignment and conflict. So when we compared these statistics between genes underlying symbiotic pleiotropy, we found significantly higher values for genes that underlie alignment, suggesting greater nucleotide diversity as well as a stronger signature of balancing selection, although the latter could also be due to recent population contraction. So we probably need to do other types of statistics to tease those scenarios apart. But what was interesting is that we basically didn't find the same pattern when we compared genes underlying rhizobium fitness pleiotropy, because we didn't see any significant differences between those. So to summarize, although these results are preliminary, the last ones especially I just showed you we're still working on, our results so far suggest that patterns of historical selection potentially differ between genes underlying conflict and alignment, whereas we did not see the same types of differences for genes underlying rhizobium fitness pleiotropy. And to conclude, I showed you how we can take naturally occurring phenotypic variation in symbiotic populations and determine the genetic basis of key symbiotic traits. And then we can use this genotype to phenotype map in order to examine the relationships among fitness proxies at both the organismal and genomic levels, which can then be used to predict how hosts and microbes potentially co-evolve. And overall, we found that fitness conflict is largely resolved in natural symbiotic populations, given the strong signature of fitness alignment that's governed by these pleiotrophic variants with concordant effects. And our results suggest that coevolution between hosts and symbionts would result in more beneficial evolutionary stable interactions, although the strength of coevolution seems to depend on the particular host microbe genotype pair interacting. We could also use this information to model how environmental conditions might modulate alignment or conflict over time, which would be useful for predicting the effects of climate change, for example. Or specifically, within the symbiosis, you could add external sources of nitrogen, such as fertilizer, or even elevate CO2 emissions to see how these factors will influence the degree of fitness conflict or alignment. And of course, these kinds of experiments are being done currently. And then finally, I want to make the point that although we're using this great model system, similar experiments could be done on a wide range of microbes, and especially those known to incur significant costs on human health or the agricultural industry, meaning that this approach could be useful for understanding how microbial evolution impacts human society more generally. All right, with that, I'd like to thank all my co-authors here, highlighted in orange, and especially my postdoc advisor, Katie Heath. I'd like to thank the Infection Genomics for One Health team, headed by Dr. Richard Whitaker. And I'd like to acknowledge funding from the Institute for Genomic Biology and NSF, and most importantly, thank my fellows, Stephen, for the invitation and organizing the symposium, and then to all of you for your attention. And I would be happy to take any questions.